Ladies and gentlemen, we just received the latest updates on the ongoing IT virus epidemic in Lamaland. It seems that the IT virus is moderately lethal, but new cases have been identified in a new city. Public health experts warned us of this possibility and are thankfully fully equipped to deal with the new Lama patients and a few humans. Researchers are also working hard to understand this new virus better.
The general public is worried and is asking numerous questions. Where does this virus come from? When did it begin circulating in Lamaland? Where did this epidemic start? Scientists now have access to the genome of the virus, which they can use to trace its evolution and reconstruct its geographic spread. Indeed, specimens have been taken from infected patients to get samples of the virus. They have then been processed in the lab to sequence their genome. Because the epidemic is a result of a single virus introduction, the sequences obtained are similar and relatively easy to compare. However, software for genomic analysis is essential. Genomic messages of viruses might be small compared to other biological entities. They are still several thousand characters long. Once the comparison is made, we can assume that the most similar messages are probably close in the transmission history of the virus. By using this principle, and our knowledge on how viral genome change, with certain changes being more frequent than others, specialists reconstruct a kinship tree. This tree is a way to represent the hypothetical evolutionary history of the virus. Is it possible to go further and use this tool to answer the question of where this new virus comes from? Yes. This approach, for example, was used in a scientific paper published in 2018 about the MERS coronavirus, to find support for the theory that camels transmitted the virus to humans. In particular, the study compared sequences of MERS coronavirus genomes in humans and camels in order to obtain a tree representing the history of the epidemic between the two species. The tree showed that most individual human samples are more closely related to samples from camels than to samples from other humans. This indicated that human MERS coronavirus sample that was separated in a tree were the result of independent transmission events from camel. These are called host jumping events and are responsible for the origin of most new viral epidemics. What about the IT virus spreading in Lamaland? We know that there is a similar virus that has been infecting humans who live close to llamas. Was the epidemic triggered by multiple human-to-llama transmission events in different cities, as was the case for MERS coronavirus? To explore this hypothesis, we can compare the differences between the viral sequences from llamas and humans. Here, we see that samples from llamas are closely related to each other, and not to samples from humans. This suggests that the IT virus is well adapted to llamas. It is a llama-only epidemic, and we need to understand it better to be able to stop it. Fortunately, the genomic sequences are not the only information that we have about the IT virus samples. We also know where and when the samples have been collected. We can use this additional information to reconstruct a more detailed history of the epidemic. By using the date of sampling, it is possible to estimate when the IT virus began circulating in the llama population. Indeed, all llamas were not infected at the same time, nor were the samples collected simultaneously. Some viral genome sequences studied emerged before others, which might favor some scenarios and cast doubt on others. As explained in the second episode of Byrevo, temporal information coupled with sequences can also inform a molecular clock that can be used to estimate the times of important events linked to an epidemic. This approach was used by researchers to estimate when the human immunodeficiency virus, HIV, arrived in the United States. Researchers compared the sequences of HIV samples collected in the late 70s in people who did not know they were infected by the virus but participated in a study on hepatitis B. The analysis showed that although the first cases of AIDS in the US were detected in 1981, the virus was circulating in the country since the early 70s. For the HEAT virus, analysis of viral genomic sequences and their sampling dates allows us to conclude that it has been circulating in Lamaland for about a year. Spatial information can complement genetic and temporal information, allowing us to go even further. We know where each viral genome has been sampled, which can help us improve our model and identify the time and place that the epidemic originated, as well as reconstruct the path of the epidemic in Lamaland. This was a strategy used by researchers in 2019 working on the rice yellow mottle virus. They showed that the virus arrived in the northwestern part of Madagascar before it spread independently on the west and east side of the island, without going into the central mountainous highland plateau. Logically, the landscape influenced how the epidemic spread. For the HEAT virus, data indicate that it appeared in the rural area A, and then reached city B, where it spread in the population. It then spread to E and then to C, separated from B by a river that probably limited movement, and thus reduced the spread of the HEAT virus between B and C early in the epidemic. Samples observed in E and C do not appear to be closely related, which is consistent with the two cities being separated by a river without any regular travel connection across it. Finally, the virus reached D, linked to C by cross-river travel that had been highly reduced but not stopped after the virus reached C. This hypothetical example shows that studying an epidemic is like an investigation. Such work is sometimes conducted after the epidemic, but it can also be done in real time, like a race against time and the virus. In any case, researchers need to combine all the available information to be able to reconstruct an epidemiological scenario as realistically as possible. The dates and locations of infection are precious information that are complemented by a new and very valuable stream of data: viral genomic sequences. Researchers can combine these different pieces of information in order to uncover how viruses evolve and spread in great details.